Hi, everybody. Good afternoon, and welcome to another session of daily lectures from New Age Cosmetic White EdTech Healthcare Consultancy. I'm Dr. Bobby. We're on the subject of molecular biology, and we're on the topic of synthesis of commercial products by recombinant microorganisms. And we're on the subtopic, as mentioned below. The subtopics will be restriction endonucleases, a lipase, small biological molecules, under which come synthesis of L-ascorbic acid, microbial synthesis of indigo, synthesis of amino acids, microbial synthesis of lycopene, increasing succinic acid production, antibiotics, cloning antibiotic biosynthesis genes, modulating gene expression in streptomyces, synthesis of novel antibiotics, engineering polypeptide antibiotics, improving antibiotic production, design antibiotics, biopolymers, xanthan gum, melanin, adhesive protein, rubber, polyhydroxyalkanoids, hyaluronic acid. And then will be the summary, followed by review questions. Now on to the topic of restriction endonucleases. The recombinant DNA technology would not be possible without a ready supply of restriction endonucleases. Now, and currently there are more than 300 different restriction endonucleases commercially available. Excuse me. And these have had worldwide sales in the range of $350 million. Just a general info, and now it should be much more. These enzymes occur naturally in many different microorganisms, including species that are aerobic, anaerobic, photosynthetic, diazotrophic, mesophilic, thermophilic, psychrophilic, and either slow or fast-growing. For each of these organisms, a detailed fermentation protocol specifying the temperature, pH, medium, composition, and oxygen tension has to be developed and optimized to achieve the maximum yield of the target restriction enzyme. To avoid having to maintain a large number of different microorganisms, stock a very wide range of microbial growth medium components, design several different types of fermenters, and spend an inordinate amount of time developing optimal growth conditions for a large number of different organisms. One major supplier of restriction enzymes lists 265 different enzymes in its catalog. Investigators often clone restriction endonuclease genes into E. coli. Exclusive use of E. coli allows bioengineers to standardize the production conditions for all restriction endonucleases. In addition to E. coli, cells grow rapidly to high cell densities, and can be engineered to significantly overexpress each target enzyme or each target restriction enzyme. Although the technology for isolating and expressing foreign genes in E. coli and some other host organisms is well established, it should be remembered that the host organism is a living entity that can be dramatically affected by the production of or presence of a heterologous protein. For example, overexpression of a heterologous protein may drain the host organism of important metabolic resources and as a result adversely affect its growth. In addition, the presence of a heterologous protein may be lethal to the host. For example, restriction endonucleases digest DNA at sites that are present at all DNA molecules. As a result, an organism that expresses a cloned restriction endonuclease gene is likely to have its own DNA degraded unless a protection mechanism is present. Microorganisms that make restriction endonucleases have evolved a self-protection system. Methylation of one or more of the bases of the DNA within their recognition sequence prevents the homologous restriction endonuclease from cutting the DNA at this site. Gram-negative microorganisms have an added mode of protection that entails localizing the restriction endonuclease within the periplasmic space and the methylation modification enzyme in the cell cytoplasm. This compartmentalization physically separates the restriction endonuclease from the DNA while ensuring that the modification enzyme has ready access to the chromosomal DNA. In addition to protecting the cell from the toxic effects of the restriction enzyme, this segregation provides a cellular defense against attack by any foreign DNA, such as DNA from a bacterial virus that might enter the periplasm. One way to circumvent the problem of host DNA degradation by heterologous restriction endonucleases is to clone and express the gene for both the restriction enzyme and its specific cognate modification enzyme in the host organism. Cloning of both these genes in the same organism is technically complex unless both the restriction endonuclease and methylation genes are close to each other on the chromosome. In addition, to prevent the digestion of the host DNA by the restriction endonuclease, it is imperative that after transformation, the methylation activity be expressed prior to the production of the restriction endonuclease. One of the first restriction enzyme genes to be cloned into E. coli encoded the enzyme PST1 from Providentia stuartii, a gram-negative bacterium. It is important to note that for a particular genus and species, only some strains encode restriction enzymes. Therefore, in this strategy, in order to easily transform the host E. coli strain without downgrading the input plasmid DNA, it was necessary to utilize an E. coli strain that was unable to synthesize the enzyme KORAH. I'll explain the diagram separately as I've done before. And the chromosomal DNA from P. stuartii was digested with HIND3 and ligated into HIND3 serum plasmid PBR322. Following the introduction of the P. stuartii clone bank library into E. coli HB101 cells, transformants were grown in liquid medium before being infected with bacteriophage lambda to test for the production of the restriction enzyme. When a restriction enzyme gene is expressed, the host cells become resistant to the lytic action of DNA bacteriophages, such as lambda, because the restriction endonuclease extensively degrades infecting bacteriophage DNA. 3 transformers that were resistant to lysis by lambda were grown, and samples were osmotically shocked to release the periplasmic proteins, which were assayed for PST1 restriction enzyme activity. Positive clones were assayed for PST1 methylase activity. One positive clone from this experiment contained within a 4.0 kilo base pair Kb DNA fragment an intact PST restriction endonuclease as methylase operon, including the P. stuartii promoter. In this construct, the natural temporal order of synthesis, the methylation enzyme preceding the restriction endonuclease was maintained. The level of the PST1 restriction enzyme expressed in E. coli was approximately tenfold higher than in P. stuartii. Excuse me. As expected, PST1 was localized in the periplasm and the methylation enzyme was localized in cytoplasm. Production of PST1 using this E. coli clone is simpler and more efficient than production with stuartii. And another strategy also has been used to isolate the genes for restriction and modification methylation enzyme systems. It was developed by a company that eventually became one of the world's leading suppliers of restriction enzymes and consists of the following steps. A clone bank was made from the DNA of a donor organism that had a previously identified restriction endonuclease. The plasmid vector had at least one recognition site for the target restriction endonuclease. The clone bank was introduced into E. coli by transformation. This step increased the amount of recombinant plasmid DNA and also allowed the expression of the modification enzyme. Plasmid DNA was isolated from transformed cells that had been grown in liquid media under conditions that selected for the presence of the plasmid. The plasmid DNA preparation was treated with the target restriction endonuclease. E. coli cells were transformed with the restriction endonuclease treated plasma DNA preparation. The rationale for this procedure is that clones that carry and express the target modification enzyme will produce plasmid DNA that is resistant to digestion by the target restriction endonuclease because their DNA will be methylated at the recognition sites. For example, following transformation of E. coli by a PBR322 HIND3 clone bank of desulfo-vibrio desulfuricans DNA, plasmid DNA was isolated and digested with the restriction enzyme DDEI. Plasmids that encode and express the DDEI modification enzymes are not digested by DDEI because the 8 DDEI recognition sites of PBr322 are methylated. After the DDEI treatment, the remaining plasmid mixture is used to transform E. coli. Only intact circular plasmids yield transformants, and these carry the gene for a functional DDEI modification enzyme. All of the plasmids are degraded by the restriction endonuclease. Their resulting transformants must then be assayed for the DDEI restriction enzyme activity to determine which clones have the genes for both the modification enzyme and the restriction endonuclease. This strategy is effective for any restriction enzyme gene that is physically close to its modification enzyme gene. Most restriction enzymes are encoded on the same operon as their cognate modification enzyme and is cloned into a plasmid vector that has at least one recognition site for the target enzyme. Now, this is a diagram which shall be explained later. And of course, I'll just give it a mention, then we'll go into the details later of the figures. Scheme for cloning the DDEI modification enzyme. Plasmids with cloned D-sulfuricans DNA are used to transform E. coli cells. On to lipase. Excuse me. Fatty stains are a persistent problem for the laundry industry. A combination of high temperature and high alkalinity can effectively emulsify and remove many fatty stains. However, these conditions often damage fabrics and also require large amounts of energy. On the other hand, the addition of lipases that are compatible with the wash conditions, such as the enzyme, produce Pseudomonas alkali genes, which may provide an effective solution to this problem. Unfortunately, this enzyme is produced at such low levels that it is prohibitively expensive to use in the cleaning of laundry. Moreover, researchers have found it extremely difficult to overproduce the enzyme in a variety of heterologous hosts, including Bacillus licheniformis, E. coli, Streptomyces lividans, Aspergillus niger, Clyveromyces lactis. The difficulty in overexpressing the P-alkali genes lipase may reflect the requirement for the simultaneous expression of another gene product that is involved in either the secretion or the stabilization of the bacterial lipase. To isolate the lipase gene from P-alkali genes as well as any other gene whose expression was linked to lipase gene expression, the enzyme was first purified. The amino acid sequence of the N-terminus was determined, and an oligonucleotide probe that corresponded to 11 amino acids from this region of the protein was synthesized. The oligonucleotide probe was used to screen a clone bank of P-alkali genes DNA, with the result that a clone that contained all of the lipase gene and a portion of the additional gene was obtained. The DNA fragment encoding the lipase gene and a portion of the additional gene was used as a hybridization probe to screen another clone bank, with the result that the rest of the second gene was isolated. The two fragments were spliced together, cloned into a broad host range expression vector and used to transform P-alkali genes. The lipase structural gene is called lip-A, and the second alpha gene is called lip-B. When the vector was derived from a low copy number plasmid, the lipase activities of the transformants were 4 to 5-fold greater than that of the wild type, regardless of the presence or absence of the second gene lip-B. However, with the high copy number plasmid, the lipase activities of the transformants were about 20-fold greater than that of the wild type in the absence of lip-B gene and approximately 35-fold greater than that of the wild type in the presence of the lip-B gene. Since the lipase is secreted into the growth medium, very little purification should be necessary before using it in laundry detergent. Rather, the cells must be removed and then the growth medium should be concentrated. However, when the production of the growth of this recombinant organism was scaled up from 10 liters to 10,000 liters, considerably less lipase activity was found than was expected. This was attributed to the production lack of removal of large amounts of carbon dioxide in the growth medium. Modification of some of the operating parameters of this large fermenter resulted in a significant decrease in the level of dissolved carbon dioxide and decreased inhibition of lipase accumulation. This work went a long way toward providing lipase of sufficient quality and insufficient quantity for use in laundry detergent. Small biological molecules. With recombinant DNA technology, it is possible to modify metabolic pathways of organisms either by introducing new genes or by altering existing ones. The goal is to create an organism with a novel enzymatic activity that can convert an existing substrate into a commercial compound that with current technology can be produced only by a combination of chemical treatments and fermentation steps. Early metabolic engineering experiments typically modified one or two genes in a biosynthetic pathway. However, with the knowledge of the complete DNA sequences of many bacterial genomes and the global information on the expression of bacterial genes derived from studies that employ microarrays, proteomics, and metabolomics, it is possible to develop strategies to improve the yield of microbially produced molecules that involve the introduction or modification of an entire panel of related genes. Right, synthesis of L-ascorbic acid. L-ascorbic acid, vitamin C, is currently synthesized commercially, but an expensive process starts in with a deglucose that includes one microbial fermentation step and a number of chemical steps. Figure 13.5 shows that. And the last step in this process is the acid catalyzed conversion of 2-keto-L-gulonic acid, 2-KLG, to L-ascorbic acid. Biochemical studies of the metabolic pathways of a number of different microorganisms have shown that it may be possible to synthesize 2-KLG by a different pathway. For example, some bacteria, Acetobacter, Gluconobacter, Owenia, can convert glucose to 2,5-diketogluconic acid, 2,5-DKG, and others, Conibacterium, Brevibacterium, and Arthrobacter, have the enzymes 2,5-DKG reductase, which converts 2,5-DKG to 2-KLG. The current procedure for synthesizing ascorbic acid could be improved by producing 2-KLG from glucose by co-fermentation with suitable organisms. Unfortunately, co-cultivation has problems of its own. For example, the two fermenting organisms might have different temperature and pM optima. The medium requirements and growth rates also might differ in such a way that the fermentation conditions are optimal for one organism and suboptimal for the other. This situation leads to the eventual washout, depletion, or loss of one of the organisms. Some of these incompatibilities may be overcome by utilizing a tandem fermentation process in which the two organisms are cultivated in succession. Of course, this approach requires two separate fermentations rather than one. If the organisms have different growth requirements, it is difficult to run the process on a continuous basis. Therefore, the best way to convert glucose into 2-KLG would be to engineer a single microorganism that carried all the required enzymes. The conversion of D-glucose to 2,5-DKG by Owenia microbicola, herbicola, it includes several enzymatic steps, whereas the transformation of 2,5-DKG to 2-KLG by Conibacterium sp requires only one. Consequently, the simplest strategy for constructing a single organism that is able to convert D-glucose to 2-KLG is to isolate the 2,5-DKG reductase gene from the Conibacterium sp and express it in E. herbicola. The first step in cloning the 2,5-DKG reductase gene from the Conibacterium sp involved purifying the enzyme and determining the sequence of the first 40 amino acids from the N-terminal end of the molecule. On the basis of the known amino acid sequence, 243 nucleotide DNA hybridization probes, each corresponding to a different portion of the protein molecule, were synthesized. Because 71% of the nucleotides in the Conibacterium sp are either G or C, the probes were designed to include, where possible, A, G, or C in the third position of all codons, thereby minimizing the extent of the mismatch between the probe and the target DNA. This approach was taken because, at the time that this work was done, mixed probes were not readily available. A Conibacterium DNA clone bank was screened with these two probes. Any clones that hybridized with only one of the probes were discarded, and it was assumed that any DNA that interacted with only one probe was probably not target DNA. A clone that hybridized with both probes was isolated and then sequenced. It contained the 2,5-DKG reductase gene. The DNA sequence that were upstream of the ATG start signal were deleted and replaced with transcriptional and translational signals that function in E. coli. Because the regulatory sequence is from gram-positive microorganisms such as Corynebacterium spp are not efficiently utilized by E. coli, this construct expressed 2,5-DKG reductase activity in E. coli and was subsequently subcloned onto a broad host range vector, which was used to transform Herbicola, which is able to use E. coli transcriptional and translational signals. The transformed Ovenia cells were able to convert D-glucose directly to 2-KLG. The endogenous Ovenia enzymes localized in the inner membrane of the bacterium were converted to 2,5-DKG and cloned 2,5-DKG reductase localized in the cytoplasm catalyzed the conversion of 2,5-DKG to 2-KLG. Thus, by genetic manipulation, the metabolic active capabilities of two very dissimilar microorganisms were combined into one organism, which was able to produce the end product of the engineered metabolic pathway. This recombinant organism should be useful as a source of 2-KLG for the production of L-ascorbic acid, thereby replacing the first steps of the currently used process. The commercial utility of the cloned 2,5-DKG reductase gene product might be improved by replacing certain amino acids of the enzyme to create mutants with increased catalytic activity and enhanced thermal stability. When the 2,5-DKG reductase gene was first isolated, the amino acid residues that contributed to the active site of the enzyme were not known. However, from the primary amino acid sequence, computer modeling predicted an enzyme structure with an 8-stranded alpha-B barrel. Figure 13.8 shows that this structure consists of 8 twisted parallel beta strands arranged close together, surrounded by 8 alpha helices that were joined to the beta strands through loops of various lengths. This folding pattern that had previously been observed for 17 other enzymes whose crystal structures were known. By comparison with the structure of these other proteins, 3 of their loops that might be involved in substrate binding were identified. Using oligonucleotide-directed mutagenesis, 12 different mutants, each with a single amino acid change in one of these loops, were constructed. Of the 12 mutants, 11 produced enzymes with a lower 2,5-DKG reductase specific activity than that of the native form of the enzyme. The 12th mutant, in which amino acid residue 192 was charged from glutamine to arginine, had approximately twice the activity of the native enzyme. Kinetic studies reveal that this increase in activity resulted from a 1.8-fold increase in the maximal rate of the enzyme catalyzed reaction Vmax and a 25% decrease in the Michaelis constant Km of the enzyme catalyzed reaction. Now, the reaction catalyzed by 2,5-DKG reductase utilizes reduced nicotinamide adenine dinucleotide phosphate NADPH as a cofactor. However, the cellular concentration of reduced nicotinamide adenine dinucleotide is usually about 10-fold greater than that of NADPH, or the financial cost of NADPH is about 10 times higher than that of the NADH. To lower the cost of the bacterial production of ascorbic acid, it would be beneficial to engineer a version of 2,5-DKG reductase that used NADH instead of NADPH. The only structural difference between NADH and NADPH is the presence or absence of a phosphate group attached to the 2' side of the adenine moiety. From the 3-dimensional structure of 2,5-DKG reductase complexed with NADPH, it appears that 5 amino acid residues interact directly with 2' phosphate residue of NADPH. Usually cassette mutagenesis, a total of 40 different mutants of this enzyme were constructed, and each constructed mutant 1 of the 5 amino acid residues that computer model suggested interacted with the 2' phosphate residue was changed to a different amino acid. Following the expression, purification, and kinetic characterization of the 40 mutants in E. coli, it was observed that changing 3 of the 5 selected amino acids resulted in increases in 2,5-DKG reductase activity with NADH as a cofactor. In the best case, when the arginine residue at position 238 was charged to histidine, there was a 7-fold improvement over the wild type with NADH as a cofactor. Moreover, after 2 amino acid alterations were combined in one protein enzyme that showed even more activity with NADH included a change of the lysine residue at position 232 to glycine, as well as a change of arginine at position 238 to histidine. Also, when the best NADH active mutant was combined with the double mutant which increased the binding of the substrate. Even further improvements in activity with NADH were observed. The activity of the enzyme isolated from the final construct was 72 times higher than the activity of the wild-type enzyme. It now remains to be seen whether this engineered enzyme can be used as the basis for the economically efficient biological synthesis of ascorbic acid. The microbial synthesis of indigo is the next subtopic. A number of bacteria, now most notably Pseudomonas spp, have the ability to use a variety of organic compounds such as naphthalene, toluene, xylene, and phenol as the sole carbon source. In many instances, the genes encoding the enzymes for the degradation of these organic compounds are located on large, naturally occurring plasmids, typically 50 to 200 kb in size. Research on these bacteria often requires detailed genetic and biochemical studies so that the genes encoding enzymes catalyzing important steps in the pathway can be treated for modification. Occasionally, despite the original purpose of a study, an unexpected but useful discovery is made. For example, plasmid NAH7 has two separate and distinct operons that allow pseudomonads that contain this plasmid to grow on naphthalene as the sole carbon source. As a first step toward characterizing these genes, NAH7 plasmid DNA was digested with HIND3, and the fragments were ligated with the linear HIND3 digestive plasma PBR322. This clone bank was introduced into E. coli cells, and transformants were selected on the basis of their resistance to ampicillin and sensitivity to tetracycline. All the transformants were then tested for the production of non-volatile metabolites that might result from the hydrolysis of radiolabeled naphthalene. During the characterization of one of the transformants that had a 10.5 kb insert and could convert naphthalene to salicylic acid, it was observed that when the minimal growth medium contained a tryptophan, it turned blue. A thorough analysis of the blue color revealed that the transformed E. coli cells were synthesizing the right dye indigo. This synthesis was achieved in 4 steps. Conversion of tryptophan in the growth medium to indole by the enzyme tryptophanase, which is produced by the E. coli host cell. Oxidation of indole to cis-indole 2,3-dihydrodeol by naphthalene dioxygenase, which is encoded by the DNA that was cloned from NAH7. Spontaneous elimination of water, air oxidation to form indigo. Thus, the combination of enzymes from 2 different pathways and 2 different organisms resulted in the synthesis of an unexpected compound, the dye indigo. In addition, introduction of the gene for the enzyme silane oxidase, which is encoded in TOL plasmid, can convert tryptophan to indoxyl, which then spontaneously oxidizes to indigo. And indigo, a commercially important blue pigment that is used to dye both cotton and wool, was originally isolated from plants, but is currently synthesized chemically. At present, approximately 13 into 10 ways to 6 kilograms of indigo worth more than 250 million is produced every year. Indigo, the coloring agent in blue jeans, is the largest selling dye in the world. The ability to produce indigo from bacteria opens the possibility of developing an efficient and economically commercial microbial process for its production. This process would avoid the use of hazardous compounds such as aniline, formaldehyde, and cyanide, which are needed in the chemical synthesis of indigo. Despite the environmentally friendly nature of engineering bacteria to make indigo, at the present time the chemical synthesis of the dye is less expensive, thwarting commercial schemes to produce indigo biologically. One approach to improving indigo production in E. coli involves engineering the host strain to overproduce tryptophan, the raw material for the process. However, despite improved efficiency when a tryptophan overproducing E. coli strain is used, the overall process requires additional improvement before it is economically competitive with chemical synthesis. Synthesis of amino acids. Amino acids are used extensively in the food industry as flavor enhancers, antioxidants, and nutritional supplements. In agriculture as field additives, in medicine infusion solutions for post-operative treatment, and the chemical industry as starting materials for the manufacture of polymers and cosmetics. Synthesis of amino acids. Amino acids are used extensively in the food industry as flavor enhancers, antioxidants, and nutrition supplements. In agriculture as feed additives and medicine infusion solutions, or for post-operative treatment, and in the chemical industry as starting materials for the manufacture of polymers and cosmetics. Table 13.1 shows that it is estimated that more than 2.5 million tons of amino acids worth more than $9 billion were produced worldwide in 2008. L-glutamic acid, which is used in the manufacture of the flavor enhancer monosodium glutamate. Remember this point? It could come for the exams. Make up around half of the total volume. That's about $4 billion. So table 13.1 shows the amino acids and applications. Please take a screenshot and that should help you out. Just for a mention of the major applications will be alanine flavor enhancer, arginine therapy for liver diseases, aspartic acid flavor enhancer, sweetness synthesis, asparagine diuretic, cysteine is bread production therapy for bronchitis, antioxidant, glutamic acid is flavor enhancer, glutamine therapy for ulcers, glycine is sweetness synthesis, histidine is therapy for ulcers, antioxidant, isoleucine in IV solutions, leucine IV solutions, lysine, feed additive, food additive, methionine feed additive, phenylalanine in infusion sweetener, synthesis, proline is intravenous solutions, serine, cosmetics, threonine is feed additive, tryptophan intravenous solutions, antioxidants, tyrosine intravenous solutions, precursor for L-dopa, and valine for IV solutions. For the most part of amino acids are commercially produced either by extraction protein hydrolysis or fermentation products of either Corynebacterium or Brevibacterium spp, which are both non-sporulating gram-positive soil bacteria that segregate large amounts of amino acids into the growth medium. And traditionally, the productivity of these organisms has been improved by mutagenesis and subsequent screening for strains that overproduce certain amino acids. However, this way of developing new strains is slow and inefficient. By using detailed biochemical information about the enzymes that are involved in biosynthesis of various commercially important amino acids, it is more expeditious to isolate and manipulate the specific genes encoding the key components of a particular pathway. However, this type of genetic engineering is not a simple matter. For example, the pathways leading to the biosynthesis of certain amino acids contains a number of different enzymes, each of which may either be activated or inhibited by a number of metabolites present in the cell. This makes it difficult to know which enzymes to manipulate in order to enhance the yield of the product. Because the most broad host range plasmid vectors replicate only in gram-negative organisms, it is necessary to construct expression vectors that are specifically suited for Corynebacterium and Brevibacterium spp. Such cloning vehicles might take the form of E. coli Corynebacterium shuttle vectors. The E. coli portion of the plasmid could encode resistance to the antibiotic tetracycline, chloramphenicol, or kanamycin. Because both E. coli and Corynebacterium spp are susceptible to these antibiotics, they could be used as selectable markers in both organisms. An efficient transformation protocol for Corynebacterium glutamicum. The species of Corynebacterium often used in these experiments is still needed. Also, many C. glutamicum genes are not efficiently expressed in E. coli. Therefore, for selection schemes that depend on gene expression, example complementation, the anti-clone bank should be transformed into C. glutamicum. Unfortunately, the transformation frequency is very low when DNA is introduced into C. glutamicum by either directly transformation or electroporation. However, effective transformation of C. glutamicum is achieved when foreign DNA is introduced by conjugation or after the formation of protoplasts, that is, after removal of the cell wall with lysozyme. The transformation of protoplasts is made possible by adding polyethylene glycol to facilitate the uptake of exogenous plasmid DNA. Some progress has been made in increasing the amino acid output of glutamicum, C-glutamicum for example. The synthesis of the essential amino acid tryptophan was enhanced by introducing it into wild-type C-glutamicum cells a second copy of the gene encoding antranilite synthetase, which is the rate-limiting enzyme in the normal tryptophan biosynthetic pathway, as shown in figure 13.11. The following protocol describes one way to isolate the antranilite synthetase gene. A library of Brevibacterium flavum chromosomal DNA is cloned into a C-glutamicum E. coli shuttle vector and introduced into a mutant strain of C-glutamicum to produce the no active antranilic acid synthetase. The mutant strain was unable to grow on minimal medium unless antranilic acid was added. Therefore, transformants were selected by the ability to grow in the absence of antranilic acid. The vector carrying the antranilic acid synthetase gene was then transferred to a wild-type strain of N-glutamicum. The amount of tryptophan produced in the mutant and wild-type C glutamicum strains, one without and one with the vector carrying the cloned antranilic acid synthetase gene, was measured. The cloned gene did indeed restore most of the capacity of the mutant to synthesize tryptophan. Moreover, the effect of adding this gene to wild-type C glutamicum was much more dramatic, with the synthesis of tryptophan being increased by approximately 130%. This level of overproduction reflects more efficient utilization of available precursor material and thus by cloning an additional gene of an amino acid biosynthesis pathway into an organism, it was possible to generate much more of the end product. An even higher level of tryptophan production was achieved when modified genes for three key enzymes, 3-deoxy-dearabinoheptulosonate 7-phosphate synthase, antranilate synthase, and antranilate phosphoribosylate transferase were introduced into C-glutamicum cells. The gene encoding these enzymes were mutagenized to render them insensitive to inhibition by the end product, feedback inhibition. An alternative to producing amino acids in Corynebacterium and Brevibacterium spp is to produce the E. coli, where the metabolic both pathways and procedure for genetic manipulation are much better characterized. Its relative ease of manipulation makes E. coli an attractive host organism for this sort of metabolic engineering. L-cysteine, which is one of the most important amino acids in the pharmaceutical, food, and cosmetic industries, has traditionally been obtained by extracting it from acid hydrolysates of human hair and animal feathers. While a number of microorganisms are able to synthesize L-cysteine, high levels cannot be synthesized from glucose because L-cysteine feedback inhibits the enzyme serine acetyltransferase, which catalyzes one of the steps in the biosynthesis of L-cysteine. To try to remedy the situation, the methionine residue at positive position 256 in the E. coli serine acetyltransferase amino acid sequence was systematically changed to each of the other 19 amino acids when E. coli was transformed with several plasmids carrying cis-E genes, encoding altered serine acetyltransferase. Several transformants with altered forms of serine acetyltransferase products produce higher levels of cysteine than did the wild-type enzyme. Next, plasmids encoding the most effective serine acetyltransferase derivatives were used to transform a strain of E. coli that did not degrade by L-cysteine. And the transformants included several different strategies and produced a much higher level of L-cysteine that had previously been possibly by merely manipulating an E. coli serine acetyltransferase gene. Whether the expression of this protein can be further improved and how this affects the synthesis of L-cysteine remain to be determined. To rationally engineer a bacterium to modify its metabolism so that it overproduces a particular amino acid, it is essential to understand how many of the metabolic pathways of the bacterium are interrelated and regulated. Subsequent systematic re-engineering of the metabolism of the organism can be performed to achieve a much higher yield than would ever be possible by merely modifying one or two genes in the immediate biosynthetic pathway of a particular amino acid. At each stage in the development of the re-engineered bacterium, it is possible to monitor the levels of a wide range of transcripts using microarray and metabolomics. Moreover, the mRNA and metabolite expression data allow the construction of a detailed computer model that can be used to predict the effects of other possible genetic manipulations on the production of the target amino acid, while the approach is relatively new. The rational genetic engineering may be used to engineer bacterial strains for a variety of purposes, in the addition to the production of specific amino acids. And to rationally engineer E. coli to produce L-valine, it was necessary to introduce a large number of mutations into the E. coli genome. The flux from glucose to L-valine was first improved by abolishing feedback inhibition by L-valine of the enzyme that converts pyruvate to acetolactate. Then the subunits of this enzyme were overexpressed by replacing the endogenous promoter with a strong constitutive promoter. Next, the carbon flux toward L-valine was increased. The gene for the enzyme that converts L309 to ketobutyrate was knocked out, as were the genes encoding enzymes that convert 2-ketoisovalerate to either pantothenate or leucine. In addition, some of the genes that encoded enzymes that convert pyruvate to acetolactate were amplified. Subsequently, the expression levels of the genes were encoding enzymes that convert 2-acetolactate to L-valine via 2,3-hydroxyisovalerate and 2-ketoisovalerate were all increased by modifying the regulatory regions of these genes. In addition, the LRP gene encoding the leucine response regulatory protein was amplified, since some genes in L-valine pathway were under the positive transcription control of the protein. Finally, the expression of the two genes that encoded proteins responsible for the export of L-valine from the bacterial cell was amplified, since it was thought that the low level of these proteins was limiting to L-valine production. Following the extensive metabolic engineering of E. coli, the final strain was able to produce 0.378 grams of L-valine per gram of glucose, which is higher than industrial strains of C. glutamicum that have been added by repeated rounds of random mutagenesis and selection. Moreover, researchers